Hello everyone, I am here to explain you about genome imprinting. So genome imprinting, it is said to be an epigenetic phenomenon in which the expression of certain genes is determined by whether the gene is inherited from the female or male parent. So basically this is affected by a process called as DNA methylation. So basically the epigenetic phenomenon is observed in majority of the individuals. One of the major factor for this epigenetic phenomenon is by DNA methylation. So let us study genomic imprinting with the example of two different genes. One is IGF2, which encodes insulin-like growth factor 2, and H19, which encodes an untranslated mRNA of unknown function. So you can see the location of two different genes at two different locus in a particular chromosome. So basically, when you say IGF2, IGF2 is said to be a insulin-like growth factor, which is inherited basically from the male parent. And here you can see when a deletion or knockout occurs in case of IGF2, there the male parent was seen small due to the deletion in IGF2, but the female parent were normal in size. This indicates that IGF2 is obtained from the male parent. So knockout is a phenomenon in which we can eliminate a particular gene from its function from the cell, and knockdown is a process where we can stop the expression of a particular gene at mRNA level. So in case of maternal chromosome, as I said, the second gene, which is called as H19, here you can see the basic function how maternal and paternal chromosomes are expressed into different individuals. So let me show you the differences. Here you can find IGF2 in case of maternal chromosome, it is off because it is not expressed in the female candidate. But here you can see an insulator that is present in between IGF2 as well as H19, and H19 is specific for expression in male maternal chromosome or female parent. So when there is an expression of H19, the enhancers which are located upstream to the H19 will help in the expression of H19, and they will block the expression of IGF2 with the help of an insulator which contains CKTCF, which is said to be the activator cannot act on the expression of IGF2. So in case of maternal chromosome, you can find that IGF2 is present in off state. When it comes to paternal chromosome, the enhancer which is located upstream to H19 gene, due to DNA methylation of H19, the enhancer will directly act on the expression of IGF2 and turning off the H19. So in this way, IGF2 will express only in the paternal chromosome and H19 will express in the maternal chromosome. So due to the mutation in IGF2, which causes a disease called as Beckwith-Wiedemann syndrome, which is said to be a congenital growth disorder which is seen in 1 in 15,000 births. The general characteristics of BWS is overgrowth of number of systems in the body, like born prematurely, and the baby has larger and heavier than expected time of gestation, and individuals are near the top end of the height and weight range for their age, and they will develop varieties of tumors in kidneys at the young age. And second type of genomic imprinting is said to be Prader-Willi syndrome, which occurs in 1 in 25,000 births. The individuals or babies are weak and small at birth, and they include varieties of symptoms like retardation, poor feeding caused by diminished swallowing and sucking reflexes. 12 month onwards, a pattern of uncontrollable events eating develops, which leads to obesity, and adolescents have poor motor skills and unstable humans. PWS is caused by the deletion or disruption of a gene or several genes in the region 15q11 and q13 of chromosome 15. Thank you.